Hello everyone. Welcome back to our MNS webinars. We're happy to resume our webinar series. And as you know, that the Mediterranean Neuroscience Society is serving to promote education in the neuroscience fields and to promote knowledge and increase public awareness of progress made in neuroscience field. Also to sustain scientific training and exchange and networking between the northern and southern countries of the Mediterranean. Of course, everyone outside the Mediterranean is welcome to join and welcome to be a member of our society. And we are happy today to have our president with us, Christina. How are you, Christina?
Hello. I would also like to welcome everybody and welcome our speaker for today. And thank you very much, Dr. Nermeen and Zakaria, for the organization. This is our third webinar, so you can find all the webinars also on our website. I will put on the chat as well the website of the Mediterranean Society. And also it will be a pleasure if you can make it at our meeting in Crete. It's the 10th Mediterranean Neuroscience Society Conference. I put at the chat the link. And you can see that we have opened the symposium proposal submission, and soon we'll have the submission for oral and poster abstracts in mid-December, and also we'll have some applications for travel awards for young investigators. We also got some funding from IBRO for that. Please follow the website and the social media of the MNS to be posted on that. And now about our speaker, I give it back to Nermeen. Yes.
Yeah, okay. Yeah, we're very happy today to have Carmen Medrano with us. Carmen earned her bachelor degree in pharmacy from the University of Basque Country in Spain. In 2013, she got her PhD in pharmacology from also the University of the Basque Country in Spain. Since then, Carmen earned cutting-edge experience focusing on neuropharmacology and animal behavior and neuroscience field. She received doctoral training in neuropharmacology at the University of College London. Her postdoctoral training expanded across esteemed institutions like the University of Strasbourg in France, INSERM, and the University of Bordeaux. Currently, Marie is a postdoctoral senior researcher at Neurocampus in Bordeaux University. Her research focus is into addiction and neuropathic pain and their association with the impaired cognitive functions. She combines along with her team, a variety of electrophysiological and animal behavioral techniques, and this contributed to deepen our understanding of various networks, which we will get a brief account today about the intracortical circuits. She's also, beside the scientific research, she's very enthusiastic about contributing to the scientific community in other ways, like in teaching, mentoring, and in science outreach. Marie, we are very, very happy today to have you with us, and the floor is yours.
Okay, thank you very much, Nermeen, for this very nice introduction, and thank you again for inviting me and letting me to participate in the MNS webinars. So today I would like to, I think that I can share my presentation already. Let's see if I'm able to.
Are you able to see this button with the upward arrow?
So let's see. Okay, can you see the presentation?
Yeah, we can see it.
Okay, so then today I would like to give you an overview of the project that I'm working currently in the laboratory of Marc Landry, of Dr. Marc Landry, that probably all you know, and also Dr. Thomas Michelet, both of them in Bordeaux Neurocampus. And the general project in which we are working on is called Decoding Pain and Movement using transcranial magnetic stimulation to unravel intracortical circuitry and noradrenaline's role in nociception-motor control. So let's start talking about pain. Pain, as all we know, is an unpleasant sensory and emotional experience. So on the one hand, pain is among the most important signals that our body gives us to help us to survive. If we feel that something is going to be painful, we try not to touch it. If we feel some pain, we can go to the doctor to try to diagnose what is going on. But on the other hand, pain might happen without a known cause and can persist after an injury is resolved. And in this case, pain can make people suffering this kind of pain have like a lot of difficulties to carry on their everyday activities. So we can, there are different ways in which we can classify pain. We can classify it depending on how long the pain lasts. So then we can find like acute pain, episodic pain, when the pain is coming, like leaving and coming back more or less regular or irregular periods of time, and then chronic pain when this pain lasts more than 3 months, or when it lasts when the known cause that caused the pain has disappeared. We can also classify pain depending on which is the most likely source of this pain. Then pain can be nociceptive if it is caused by, for example, an inflammation, like inflammatory pain. It can be neuropathic if it is caused by the lesion in a nerve, like, for example, sciatica. Or it can be also nociplastic if this pain involves like an awkward and an abnormal process, like central processing, like, for example, fibromyalgia. And regarding the causes of pain, well, there are multiple as well. The ones that maybe first come to our mind are the biological factors, such as nervous system sensitization, or a malfunction of immune and endocrine systems, or even genetic factors. But there are also other psychological factors, such as fear and avoidance, such as emotions and moods, or even resilience or beliefs. And there are also some social factors that can induce or prolong this pain, such as access to healthcare, economic stability, or even education. And this is why we say that pain is a biopsychological experience. So there has been and there is a lot of research working on pain, and but there is still much to learn about how and why people feel pain in different ways, even though the causes might be alike. Of course, we know more about acute pain with an obvious cause, but it's much acute pain is much better understood than chronic or neuropathic and nociplastic pain. So in general and traditionally, much research has focused on the spinal components of pain processing, and less attention has been paid to the cortical ones. However, we know that there are as many nuclei in the brain involved in pain as types of pain itself. And we usually refer to the supraspinal nuclei involved in pain as pain markers. So then pain processing involves complex interactions between sensory, motor, and cognitive functions. For example, neuropathic pain is associated with intersensory processing, but also with abnormal motor behavior. And this is the thing that I want to introduce you now, how pain and motor control are related. So something that we know is that when people experience pain, they move differently. And so then when we experience pain, we try to move in a way that is going to avoid feeling that pain. And that's normal, and that's a survival skill. However, sometimes these changes in movements persist once the pain is over. And even though pain controlling movement is something that that we know and we experience in everyday life, the neuronal interaction between pain and motor function is not yet fully understood. And this is one of the main questions of our project, right? So we can consider nociception as a continuous feedback for movement, helping the body sense and avoid possible pain or injury. Therefore, pain influences actions at multiple levels of the nervous system. As we can see here in this scheme, I'm not going to explain it like in detail, but it's a nice way to illustrate that pain affects of course, like the peripheral muscle unit is going to affect the way in which information is processed in afferent and efferent connections, of course, in how motor signals are processed in the spinal cord, and as I said, in the cortical and subcortical motor structures. So critically, there are two regions in the cortex, the primary motor cortex and the cingulate cortex, and one region in the brainstem, the locus coeruleus, that are involved in motor, sensory, and cognitive control. And the three of them have reciprocal connections, and the three of them end in a final common pathway, which is the spinal cord. So in this project, we have hypothesized that abnormal activity in this circuit may account for maladaptive pain responses and motor deficits. So then now I would like to talk a little bit about the role of each one of these different nuclei in pain. So let's start by the primary motor cortex and pain. The primary motor cortex has an essential role, obviously, in the formulation and execution of movement, and is likely to have a role in movement abnormalities. But it has been demonstrated that there is an alteration of M1 activity in chronic pain, and then this made us wonder if researchers wonder if a modification of this abnormal M1 activity could be a potential treatment for treating chronic pain, and this brings us to the introduction of the transcranial magnetic stimulation, or TMS, of the primary motor cortex. So there are several studies that have shown that the TMS of the primary motor cortex has been demonstrated to facilitate pain management by offering relief in conditions such as chronic pain and fibromyalgia. So just a very, very brief introduction of the TMS for those who are not very familiar with it. So the TMS consists in one or two coils that are going to send magnetic pulses on the brain area that is where it is located. In this case, it is located on the primary motor cortex. It is obviously a non-invasive brain stimulation that was developed in the 1980s, and since then it has been more and more used, and its use has been more and more investigated for the treatment of different neurological and psychiatric diseases. So the magnetic pulse that we induce with the TMS is going to induce an electrical current in the neurons where we apply this magnetic stimulation. But these neurons are going to send other nerve impulse to the rest of the brain and eventually to the target organs. So it has been shown that, as I said, that the TMS could give some relief for chronic pain. However, most investigations conclude that further studies are required to, one, enhance performance, two, define best brain protocol stimulations, and three, achieve long-lasting effects. Because one of the main limitations of the use of TMS in pain is that the effect, the effect on pain seems to disappear as soon as we stop the stimulation. And apparently, they claim in these studies that is the lack of neurophysiological evidence of TMS on the motor cortex, which makes the efficacy of TMS of the motor cortex for chronic pain relief not conclusive. And this is going to be one of the main objectives of our project, to try to figure out, to better explore this neurophysiological effect. So now let's have a look at the role of the cingulate cortex in pain. The cingulate cortex, as you may know, is in a powerful position to regulate the interaction between sensory, cognitive, and motor control in relation to changes in emotional and motivational states. And this is why the cingulate cortex is placed in the right place to translate intentions into actions. So there is plenty of evidence about the effect of that emotions have in the activity of the cingulate cortex, or how different types of sensory information is processed in the cingulate cortex in order to coordinate motor autonomic responses, and of course how cingulate regions are activated by pain, by noxious stimulus. So we know that the cingulate cortex is involved in pain processing. Traditionally, since the cingulate cortex is a pivotal region for emotion, plenty of studies in humans and in rodents indicate that that the cingulate cortex mediates affective chronic pain. But now we know that it is already involved in the processing of noxious stimulation, pain-induced attention, avoidance, and in the effect of endogenous opioid and placebo analgesia, and in the descending pain control. And interestingly, there is at least one study, that shows that the stimulation of the motor cortex, of the primary motor cortex with the TMS, is going to induce changes in the cingulate cortex, and these changes are going to be correlated to the magnitude of endogenous analgesic effect, which this study is very interesting to us because it's a proof that that the connections between the link between the primary motor cortex and the cingulate cortex could be a target to treat pain. Since 10 years ago, we know more and more, thanks to the work of Vogt and others, that the cingulate cortex is divided in several subregions, and these subregions are cytoarchitecturally different, and also they have different afferences and efferences. But a question that is open now is if this distinct anatomical subdivision of the cingulate cortex have specific functional and pathophysiological implications, and for that we need more functional studies as our own study. And last but not least, let's check the role of the locus coeruleus in pain. So the locus coeruleus is a very, very small nucleus in the brainstem. It's the major noradrenergic nucleus of the brain, innervating most structures of the neuraxis. And because it's like widespread innervation, the locus coeruleus is involved in plenty of physiological functions such as sleep cycle, arousal, memory, and it has been also involved. It has been its malfunction. It has been involved in plenty of in a lot of neuropsychiatric and neurological disorders, from opioid addiction to Parkinson's disease or Alzheimer's disease, right? But among all these several physiological functions that it has, we are going to focus on two. First of all, its role in arousal. Aston Jones and Cohen in 2005 describes that the locus coeruleus has bimodal activity, tonic and phasic, and that this tonic and phasic activity were very much related to the engagement of the individual in a task. So to the attention that the individual was paying in a task, and to the exploration or exploitation in a task. And this very important, the switch between phasic and tonic activity was coordinated mainly by the cingulate cortex. And this is why it's this function is very interesting for us. And in addition, the locus coeruleus has been, it is involved in nociception and spinal descending inhibitory control. As we can see here, a noxious stimulus is going to increase the activity of the locus coeruleus neurons, and this increase of activity in the noradrenergic neurons is going to release noradrenaline in the spinal cord, which is going to counteract the effect of this noxious stimulation. And of course, there are several studies, especially in rodents, which have demonstrated the effect of the locus coeruleus, the malfunction of the locus coeruleus during chronic pain. So the locus coeruleus is not only the main source of noradrenaline to the cortex and the spinal cord, but it also receives reciprocal inputs from these regions, which modulates its activity. And this is this bidirectional communication, which makes it an attractive hub for studying corticospinal processes related to nociception and attention. And well, interestingly, in the last few years, we know, thanks to a lot of research made in the last few years, we know that pupillometry can be used to assess locus coeruleus noradrenergic neuromodulation. So we know that changes in the pupil diameter may be coupled to alterations in the locus coeruleus noradrenergic neurons. And interestingly, these noradrenergic neurons of the locus coeruleus are activated by noxious stimuli, novel sensory stimuli, but also shifts in perception, which are the same stimuli that are going to be associated with changes in the pupil diameter. So based on these findings and others, we know that changes in the pupil diameter correlate directly with changes in the activity of these noradrenergic neurons. And therefore, we can use this non-invasive technique, which is the measurement of the pupil diameter, as a proxy or a measure for locus coeruleus noradrenergic activity. So now that I gave you like a view of of our regions of interest and why we are interested in them to study pain and motor control related disorders, I'm going to explain you to try to show you the objectives that we have in this project. So first of all, we would like to functionally explore the connections between the different subregions of the cingulate cortex and its locus coeruleus noradrenergic modulation. We would like to identify the cingulate cortex subregions involved in either pain, in motor control, or in both, and to test different stimulation protocols that would be effective in pain motor disorders in order to try to improve or to apply different protocols with the transcranial magnetic stimulation in patients suffering from these disorders. And we would like to investigate the cingulate locus coeruleus pathway in the sending noradrenergic inhibitory control. And now I would like to show you just a few preliminary results that I've got the last few months. So first of all, I'm going to present you the methodology, what the experiments that I performed in order to try to study these objectives. So I study the impact of the electrical stimulation of the different subregions in the cingulate cortex of the mouse in an anesthetized in mice anesthetized with isoflurane, in which I can record the pupil and I can also induce pain. So what I do is to stimulate, well, in the cingulate cortex of the mouse, they have been identified 7 different subregions. So what I do is to stimulate these 7 different subregions in each animal, and to test and to check if this stimulation induces an increase in the pupil diameter. So I'm going to show you a video in which I hope that you can see the effect of the stimulation of one of these subregions in the pupil. So here is the basal pupil, and now there is the stimulation. You see that the pupil is going to increase its diameter, and very quickly it's going to become to go back to the basal state. So in case the video was not very clear, so this is how it looks like, how the pupil looks like in before the stimulation. And we have to, this little circle is what's going to delimit the pupil of the mouse. And then this is how the pupil looks like immediately after the stimulation of the cingulate cortex. And then, as I said, between 2 or 4 seconds after the pupil is going to go back to the basal state, and then we can start stimulating it again. So the results that I got for now is that the stimulation of different subregions of the cingulate cortex in the mouse are going to induce different pupil-evoked responses. So as we can see here in this example, each one of these lines, the different color, represents the stimulation of a different subregion of the cingulate cortex in the mouse. So as we can see, the stimulation of each region is going to induce an increase in the pupil diameter. But interestingly, we see that there are significant differences between the different subregions. So then the cingulate subregions that are more anterior are going to induce a bigger pupil dilatation than the ones that are more posterior, except for the posterior ventral area, the 24A. So these results, which are in line with a study that the PhD student in the lab, Vianney Salvi, performed in rats, and that work is already submitted for publication. This suggests that the different subregions of the cingulate cortex in the mouse would be connected in a different way with the locus coeruleus, and this is why we obtain they are neuromodulated by noradrenaline in a different way. So in order to corroborate that this effect that we saw on the pupil after the stimulation of the cingulate cortex was actually due to the noradrenaline release from the locus coeruleus, I did a set of experiments in which I injected for 3 days some mice with the specific noradrenergic, locus coeruleus noradrenergic neurotoxin DSP4 or saline. So in saline, this is an example of an animal injected with saline, and as we can see, the stimulation of the different subregions of the cingulate cortex was going to induce different pupil response. However, in the animals injected with DSP4, we saw that this effect was blocked. And then what I did in another set of experiments, it was, of course, to check the effect of a noxious stimulation, in this case pinch, to the pupil diameter, to see if noxious stimulation had an impact, had a pupil response in the anesthetized mouse. So as you can see here, the pinch of the paw induced a big increase in the pupil diameter in animals injected with saline. However, in the animals injected with the noradrenergic neurotoxin DSP-4, this effect was completely blocked. So then these very few experiments confirmed that the anatomical differentiation of the cingulate cortex in mice has actually functional implications, and that the use of pupillometry, that we can use pupillometry to functionally study the locus coeruleus neuromodulation in noxious responses. So once that the protocols were established and that we saw that pupillometry was actually working in mice, then we started our second study. So in this study, which is still like the experiments are still ongoing. We want to use the pupillometry to evaluate the noradrenergic descending inhibitory control in attention deficit hyperactivity disorder, or ADHD. So why ADHD? Well, we know that ADHD, the main symptoms of ADHD are inattention, hyperactivity, and impulsivity. But we also know that ADHD is linked to chronic pain. And actually, we find a higher frequency of ADHD among patients suffering from chronic pain, especially if patients suffering from fibromyalgia, but also children suffering from chronic pain are more present more frequently ADHD troubles. In a study from the lab that it was published a couple of years ago, using a ADHD mouse model that I will explain a little bit, I will explain later on, they also show that the ADHD-like mice show hypersensitivity to mechanical stimulation, like these results went matched very well with what we see in humans. But in addition, and related to our project, we know that ADHD involves abnormal noradrenergic modulation. The strongest evidence in support this abnormal noradrenergic modulation in ADHD comes from the pharmacological treatments, because there are a few noradrenergic related treatments used for ADHD that, in addition, they can be used as well for chronic pain. And recently, there are more and more studies in which they use pupillometry. And pupillometric studies show how children suffering from ADHD show a larger pupil diameter in resting conditions, and how the pupil abnormally responds to environmental stimuli or to cognitive tasks. So in a recent study in which they use an ADHD model for rats, they show how these ADHD rats develop acute pain, develop pain chronification, and they suggested that this may be due to a dysfunction on the spinal noradrenergic releasing processes. So we have enough evidence to see to know that ADHD involves an abnormal noradrenergic modulation. However, the exact mechanism of this neuromodulation in ADHD is not well known, and this is going to be another one of our questions. But the link between ADHD and our project is even stronger, because we know that the anterior part of the cingulate cortex, the ACC, is central to both pain and ADHD disorder. Actually, a work from the lab show that the ACC is involved in nociception control dysfunctions in our mouse model of ADHD, and that ACC excitatory neurons are going to amplify nociceptive sensitization in these mice, in the ADHD mice. So then it is clear that there is a link between the neuronal substrate of ADHD and that of chronic pain. In both syndromes, we find higher pain sensitivity, but also alterations in the anterior singular cortex excitability and changes in the noradrenergic, in the locus coeruleus noradrenergic activity. So we might hypothesize that an impairment in this locus coeruleus noradrenergic neuromodulation of the anterior singular cortex regions may be associated with disrupted descending inhibitory control, exacerbating pain perception in ADHD patients. So in order to study this, we use our ADHD mouse model, which consists in an injection of the dopaminergic toxin 6-hydroxydopamine at P5 when they are newborns. So these surgeries, this lesion in the lab are carried out by Sandra and Sarah that very, very kindly are teaching me how to perform them. So the idea, the surgery consists in injecting, as I said, the toxin 6-hydroxydopamine in the ventricle of the baby mouse, and then in the adulthood, they have shown that in the adulthood, these animals show hyperactivity, inattention, impulsivity, and increased pain sensitivity. So until now, we have used, I'm still using the mechanical pain threshold measurement to establish this hypersensitivity to pain in our ADHD mice. For those who are not familiar with this test, with the von Frey test, this is a test that allows us to assess mechanical pain. So the idea is that we have a set of different filaments, and each filament has different thickness, and we are going to measure the pressure that we apply to the animal, to the paw, in grams. So we put the mouse in a cage, and the bottom of the cage is a net that is going to allow us to go through it to pinch or to press the paw with this filament. So when we see that the animal withdraws the paw, it means that that sensation is unpleasant, that is painful, and then that filament is going to establish the pain threshold. So what I did in my animals, it was to check the mechanical pain threshold in ADHD mice and in controls, and as it has been shown before in the lab, both females and males ADHD are going to show hypersensitivity to mechanical stimulation. But in addition to, as I said, in addition to establish the mechanical hypersensitivity in this ADHD model, we are very much interested in studying the diffuse noxious inhibitory control, or DNIC. So for those who are not familiar with it, the DNIC is a normal physiological process involved in pain suppression. So it's a phenomenon whereby one noxious stimulus can inhibit the pain response to another. So, in other words, is how pain inhibits pain, and it is produced by endogenous analgesia. If I may give you an example, for example, we are cooking and we burn our right hand, and immediately after, unfortunately, we pinch our left hand with a fork. We are going to feel less pain in the left hand than if we hadn't burned the right hand in the first place. So, yeah, as I said, it's a physiological mechanism that is going to protect us to suffer excessive pain, right? So we know that the NIC is induced by the inhibition of wide dynamic range neurons in the spinal cord, and it is believed that this inhibition comes from supraspinal structures, but the exact mechanism of action or the exact modulation is not well known. What it is known is that the NIC is used frequently to quantify central pain sensitization in chronic pain patients, and an inefficient NIC is considered as a risk factor for developing chronic pain and pain like diffuse syndromes such as fibromyalgia, irritable bowel syndrome, migraine. And then we are wondering if maybe the hypersensitivity to pain that the ADHD patients suffer, it would be related to an inefficient DNIC process. So in order to answer to this question, Sandra developed a protocol in which she measured the NIC in mice. So for this, what she did, it was to apply this bone freight that I talked about earlier on, to measure the mechanical threshold in the mouse. And once that mechanical threshold for pain was established, she put the tail of the mouse in hot water, and immediately after, she measured again the mechanical threshold in the same mouse. So what she saw is that in control conditions, in sham here, both females and males, they show an increased ratio in mechanical anodinia. So the threshold for the second stimulation was higher than the first one. So which means this increased ratio means that these animals had a normal DNIC function. They didn't feel that the mechanical stimuli was so painful after having the tail in the hot water. However, when she performed these experiments in ADHD mice, she saw that this technique didn't exist. This animal didn't have this physiological process, and in these animals, the threshold for a mechanical noxious stimulus, it was the same before and after having the tail in the hot water. So what I did was to adapt Sandra's protocol to my experiments in order to explore the possibility if an impaired noradrenergic modulation may underlie this ineffective technique in ADHD mice. So for this, I perform, so as I said, the experiments in mice anesthetized with isoflurane, in which I could record the pupil and apply this painful stimulus. In my case, the painful stimulus that I applied, it was a food shock. So I could apply like different intensities of food shock until I saw an increase in a pupil response, right? And that pupil response meant that the food shock of that at that intensity was painful, was noxious. So I did that, and after I put as Sandra did with her animals, I put the tail of the animal in hot water, and immediately after I apply again the food shock to establish the threshold, the DNIC threshold, right? So, well, first of all, what I saw is that in this like very first set of experiments, is that in control animals or sham, the intensity that of the food shock that I need to apply to see a pupil response is higher than in ADHD, which means that even in anesthetized conditions, the ADHD pupil reactivity the intensity of the food shock that I need to use in the ADHD animals to have a pupil reaction is lower, which matches very well with the results, the bone-free results. And it corroborates the fact that the ADHD mice feel more pain, they show a hypersensitivity to noxious stimulus. And then I applied the DENIC protocol. So indeed, these are the results for the control animals. So here, as you can see, the intensity of the food shock in basal conditions, or the intensity of the food shock after the DENIC, so after putting the tail in hot water, is higher. So it means that these control animals, they show a DENIC because they feel less pain than before the DENIC, than before putting the tail in the hot water. However, in the ADHD animals, I saw that there is no increase in food shock induced pupil response, and therefore the DENIC was also blocked, as Sandra saw in her set of experiments. So this corroborates that this DENIC is impaired in ADHD mice, pointing to the noradrenergic descending control as a target for the comorbidity between pain and attentional disorders. And in the current experiments that I'm performing these days, what I'm investigating is if the locus coeruleus neuromodulation of the cingulate cortex in the ADHD mice is also impaired, because that will give us an idea if this noradrenergic neuromodulation is impaired already at the cortical level. And well, this is that's all what I can present for now. So I would like to thank my two teams, the purinergic neuroinflammation and brain disorders team, especially Professor Mark Landry, and the ADHD team, Sandra and Sarah. And also I would like to thank my other team, my motor and cognition team, Mokoko in INSEA, especially Thomas Michelet, who is the other co-director of my project, and Vianney Salvi, who has performed the experiments in rats. And well, thank you to the GPR for the funding, for my salary, and for the project. And thank you all for your attention. I don't know if you have some questions.
Marie, thanks a lot for this interesting session. It was really interesting, and I'm sure our participants will have some questions to ask.
Maybe I can ask first. Can you hear me well?
Yeah.
Okay, thank you. It was very interesting. Very congratulations for the very important data, and I'm curious to see what's next. It's very, very interesting. And I didn't know the link between ADHD and pain. It's something that I didn't know. Of course, I work on sex difference, so maybe I'm interested in that. I saw that in some cases there were both males and females, but I know there are also sex differences in pain response. And especially for the last study with the food shock, was that on male animals?
Yeah, and I have to say, so since I started the project not a long time ago, and as every project that starts takes a while to set up the experiments, especially the pupillometry, because it's something that it was new for me and for the lab in mice. So then the results that I show you for now are only males. I have performed also the same, about the ACC reactive, the pupil reactivity after the cingulate cortex stimulation. I have those data in females, but I haven't had time to analyze yet. And related to the DENIC and ADHD, so well, it's the same. For now, for the Bonfrey, for the Bonfrey test in non-anesthetized animals, we have plenty of evidence in the lab, and myself as well, that the technique happens in both. But it's true that that females are a little bit, in like naive females, seems to be more hypersensitive than males, like in control conditions. But for the technique and for the pupillometry, is something that I have to analyze, and I want to analyze as well, because the locus coeruleus has been shown to react different, like not only to pain stimulation, but also to, for example, attentional tasks, etc., between males and females. So it's something that for now I haven't analyzed, but we are planning on analyzing yet, like to see if there are differences.
Okay, thank you. Yes, that will be very interesting and to see. Thank you. I don't know if we have any questions from the viewers. I'm not sure. They can write on the chat, apparently.
Yeah, of course. Yeah, they can use the chat. I think
because when I entered the call and I was just a viewer, I couldn't write on the chat, but maybe now it's activated. Before I couldn't. But maybe
Gopta can help us with this. I don't have the option or the availability.
Can someone from the viewers, Luisa, maybe, or someone else send us a message to see if it works? To see if we can get messages from the viewers. I'm not sure.
Luisa is a contributor, so she can write, and I think, yeah, everyone now, I think, can write.
Okay.
Yeah.
Yeah.
So, Mary, yeah, have you tried these mechanisms with the chronic pain, or just for the nociceptive pain and mechanical pain?
Yeah, so for now we have tried only with acute pain, but of course that's one of the main questions of the project, is to figure out how this system is modified, because we hypothesize that it's going to be in chronic pain. So we are planning on using the cuff model. I don't know if you know it, established by Ipekias in Strasbourg, and we are looking forward to doing those experiments. But before I would like to try to finish the ADHD as soon as possible. But of course, the chronic pain would be, because it's like implications are much wider, like in terms of mood and other disorders, and also related to motor control.
Yeah, of course. Yeah, but I have to say that the breakthrough in the ADHD pain correlation is it's very interesting to know about it, and maybe the transcranial stimulation or the magnetic stimulation will also help with the control in the noradrenaline role and the inhibition. Yeah.
Well, that's something that we are investigating in the lab. Already in humans, not in patients, but in control humans, and we are trying to set like the appropriate protocols with the TMS to see if we can see a pupil reactivity already in control. And it seems to be working well, but we still have to optimize the protocols, and this is why it's so important to perform like some some experiments in mice first, to see if one of the regions would be like more related or more likely to induce this pupil reaction than others.
Yeah. So maybe it will be more available with more targeting to this specific region, or?
Yeah, that's what we think, because at least taking into account what we have seen in mice and in rats, and this is what we are trying to study in humans as well. Some regions of the cingulate cortex will be more related, more connected, or connected in a stronger way with the locus coeruleus, and then maybe modulating specifically, targeting specifically these regions, it would be more beneficial to other, to treat like pain disorders related, or ADHD pain related to noradrenergic deficit.
Yeah. Another question about the technique. And how long does it last? How long does it last with the pain inhibiting pain?
Yeah.
Yeah.
In rodents, it doesn't last. It doesn't last much. It's just, I mean, actually, you need to apply the stimulus like very, very quickly, because I would say that in 5 minutes the effect it's over. So it's really a very efficient mechanism for the body to not to feel excessive pain, but of course it's going to disappear like more or less quickly, at least in I'm talking about normal conditions, not about chronic pain or neuropathic or ADHD. Well, in ADHD, which is very obvious and very interesting, is that this technique seems actually not to work.
Yeah. Thanks a lot. I think, does anyone would like to ask about anything? Okay. Thanks a lot, Marie. It was really a pleasure to have you with us today.
Thank you very much. I hope that I can see you in Crete with a full story about this technique, and then meet you in person. Thank you very much again for inviting me.
Thank you. Thank you.
Thanks a lot.
Have a nice day.
Hasta luego.
Hasta luego.
Bye bye, Mary.
Bye.
Ciao.
Bye. Thank you,
Ciao.
Liana.
Ciao.
Okay, thanks.